Welcome to Lecture 19, where we will go over the process of embryogenesis. Amniotes, such as mammals, reptiles, and birds, have 4 extraembryonic membranes to help support the embryo. This is considered an adaptation to terrestrial living. These extraembryonic membranes include the chorion, which is the site for gas exchange, the amnion, which is a water-filled sac that protects the embryo, the allantois, which will form the umbilical cord in mammals to help transport gases, nutrients, and wastes to and from the embryo, and it will eliminate waste in reptiles. And finally, the yolk sac. The yolk sac will enclose the yolk in reptiles and eggs, and also serves as the site for early blood cell formation in mammals. Extraembryonic layers are present within shelled eggs, and they also develop within the uterus of marsupials and eutherians or placental-bearing mammals. These layers are considered to be a key adaptation that allows for terrestrial living. Note that these extraembryonic layers are not present in other vertebrates such as fish and amphibians. This is because they reproduce in aquatic environments where nutrient and gas exchange can occur freely. The next process in embryogenesis is organogenesis. Organogenesis occurs when the embryonic germ layers begin to develop into rudimentary organs. Typically, 2 or 3 germ layers are involved in the formation of an organ. Cells can migrate to other locations in the embryo. As well, these cells may change shape or differentiate as they become specialized. We could take a look at the early formation of organs in the embryo to the right. Note the early rudimentary structures such as the eyes, the somites, as well as the tail bud. Neurulation is a process that occurs during organogenesis. Neurulation refers to the process that leads to the formation of the brain and spinal cord in vertebrates. It involves induction process. Induction occurs when a group of cells can influence the development of another group of nearby cells. In this instance, the notochord forms from the dorsal mesoderm. These mesoderm cells will then secrete signals that cause epidermal cells to form a neural plate. In addition, ridges will form on either side of the plate called the neural folds. As the cells begin to change shape in the epidermal plate, the neural tube will form along the anterior-posterior axis of the embryo. The neural plate cells will begin to fold inward and eventually pinch off to form the neural tube. This will then lead to the development of the brain and spinal cord. The notochord eventually will disintegrate. However, discs will remain as seen between the human vertebrae. In addition, during organogenesis, some cells will migrate to different regions within the embryo. This can be seen in neural crest cells as well as the cells that form the somites. Neural crest cells, if you recall from the previous slide, originate from the closing of the neural tube. When the neural tube closes, the neural crest cells will be left between the neural tube and the epidermal plate. These cells will migrate to other parts of the embryo and eventually form peripheral nerves as well as skull bones and teeth. Mesodermal cells that lay on the sides of the notochord may also separate and form somites. Some of these cells will form mesenchyme cells, while others will form vertebrae as well as muscles. The process of organogenesis in chicks is very similar to what we see in the frog. There will be pinching and infoldings, which form a 3-layered tube as seen to the diagram to the left. This 3-layered tube will be joined to the yolk at the midpoint. By 56 hours of development, rudimentary structures such as the brain, eye, as well as heart are apparent. In insects, neural tissue will form along the ventral side of the embryo versus the dorsal side as we've seen in vertebrates. There are also many cellular changes that occur during embryogenesis. Cellular changes can include changes in morphology, migration, as well as programmed cell death. These processes help to organize the various structures and tissues within the embryo. For instance, the reorganization of actin filaments will change the shape of the neural plate ectoderm cells. It changes the cells from cuboidal to wedge-shaped. This change in shape allows for the invagination of cells and overall formation of the neural tube. Cell migration is also an important part of embryogenesis. The cytoskeleton adhesion proteins that are present between adjoining cells and various components of the extracellular matrix all play an important role in allowing for cells to move within an embryo. This occurs in the movement of neural crest cells as well as in the production of somite cells. And finally, programmed cell death can also help organize the embryo during development. Apoptosis, or programmed cell death, of cells as well as groups of cells can play an important role in diversity that we see in adult forms. For instance, apoptosis is a very important process that allows for the elimination of the tail in tadpoles when the tadpole is undergoing metamorphosis to become an adult. It is important to understand how cell fate is controlled within this process of embryogenesis. Determination refers to the commitment of a cell in a particular fate pathway, while differentiation refers to its specialization in structure and function. Fate maps are important in depicting the outcome for certain regions or certain cells from an early zygote to a late-stage embryo. The developmental potential of a cell in general becomes more limited as it ages through the developmental process and as it becomes specialized or as it differentiates. Take a look at the two fate maps below. Note the distribution of endoderm, mesoderm, and epidermal cells between a blastula and an embryo in the neural tube stage. Also note the development of the tunicate. Note the presence of the blastomeres in the early embryo, and note their overall location in the larvae. Signaling molecules are also important in relaying positional information to other embryonic cells. This concept is important in establishing the overall body plan for the animal. Many of these groups of cells are present within the dorsal lip of the blastopore in amphibians, the apical epidermal ridge in vertebrates, or the zone of polarizing activity also in vertebrates. We will now take a look at the role of these locations in axis formation. If you recall in the frog embryo, the point of sperm entry helps to polarize the egg. This will later then determine the anterior to posterior axis. In general, the animal pole forms the gray crescent and marks the dorsal side of the animal. In mammals, however, the site of nuclei fusion and the site of the second polar body formation from continued meiosis in the egg helps to establish the cleavage plane, thus helping to establish the overall axis for the embryo. In chicks, the pull of gravity on the yolk cells and pH differences become important in establishing the anterior to posterior axis. This occurs as the egg travels down the oviduct. In insects, like the fruit fly, gradients of transcription factor expression, such as the gradient of BCD, NOS, HB, and CAD genes, will help to establish the anterior-posterior axis as well as the dorso and ventral axis, as seen in the diagram to the right. Once the axes, the anterior-posterior and dorsal-ventral axes are established, at that point the left and right axis of the animal becomes fixed. In addition to the dorsal lip of the blastopore, there are two other regulatory regions that have been discussed, the apical ectodermal ridge and the zone of polarizing activity. These two zones are regulatory regions that help to determine the pattern of formation in the vertebrate limb. The AER, or apical ectodermal ridge, is a thickened area that is present on the edge of a limb bud. This will help to promote the growth of the limb along the proximal and distal axis. The AER grows due to the secretion of FGF, or fibroblast growth factor, from the AER cells. The zone of polarizing activity, or ZPA region, consists of a block of specialized mesoderm that helps to regulate the limb growth on the anterior to posterior axis. Adjacent cells to the ZPA region will form posterior structures, while the further cells will form anterior structures. The diagram here depicts the difference between the AER and ZPA zones. Also note the presence of the various axes, anterior to posterior, proximal distal, and ventral to dorsal, in relation to the formation of the limb. And finally, recent research has suggested that the presence of monocilia plays a very important role in determining cell fate. Monocilia are stationary or motile primary cilia that are present in nearly all cells. Monocilia, as seen here on notochord cells, tend to grow in length during the process of somatogenesis. Motile cilia, as seen in the diagram to the right, help to generate fluid flow. This will help direct calcium ions that aids in cellular signaling as well as cellular differentiation. In general, the stationary monocilia help to act as antennae. They act to receive signals from the surrounding environment, whereas motile monocilia help to create a current flow. This will help to establish the bilateral or left to right symmetry in an animal. As a rule, the presence of monocilia, both stationary as well as motile, plays an integral role in the overall determination of cell fate.